Professor Arup Chakraborty is actually the visiting professor of IAS HKUST. So he is currently the Jockey Haslam Professor of Chemical Engineering at MIT, and he is also a professor of chemistry, physics, biological engineering at MIT. I guess this multiple affiliation give him the opportunity to attract students from various backgrounds to join this group. And that also highlights the actually underlying spirit of his multidisciplinary research activities. Actually, before he joined MIT in summer 2005, he was actually a chair professor in Berkeley. He was one of the endowed chair professors at Berkeley, and he served as department chair at Berkeley's Department of Chemical Engineering. And he was also served as the head of theoretical computational biology lab in Lawrence Livermore lab. So right now he's actually most of his research activity was actually trying to use computational theoretical strategy and principles to improve the understanding of medicine, life science in particular, in immunology, and through the understanding, then he can contribute to the design of vaccines. I think this is very much the key spirit of his research activities, and he has received many different awards. Although Arup's traditional training was in catalysis, he was able to be awarded, recognized by NIH. He received NIH Pioneer Award many years old, and his research in this area has been published in Nature, Science, PNAS. A couple months ago, he actually has a paper published in Cells on the signaling pathway from very much engineering analysis point of view. So I think this is the second or third IAS talk he did. And every time I listen to his talk, I see new assignments of these areas. So I believe that we will feel the same way today. So without further ado, please join me to welcome Professor Arup Chakraborty to this presentation.
Thank you very much. I'm pleased to be here, as usual. So as Yiming mentioned, all my work is focused on understanding various aspects of the immune system. And by way of introduction, let me say that we have an adaptive immune system which enables us to mount pathogen-specific responses against a diverse and evolving world of microbes. This is remarkable because it's not specificity to some fixed slate of pathogens, but specificity to pathogens that may not have evolved when you were born. To roughly see how this works, consider infection with a virus. So when a virus infects the host cell, it hijacks the transcriptional machinery of the host cell, thereby enabling synthesis of new viral proteins and the assembly of many new virus particles that can go on to infect other cells. So important players in adaptive immunity are B cells. We each have many clones of B cells, each with its own distinct receptor, called the B cell receptor. And if the receptor on the surface of this particular B cell can bind strongly to the spikes on the surface of this particular virus, it can get activated and then proliferate, make many copies of itself, and ultimately, by a process that I will describe hopefully a bit later in the talk, it can secrete this receptor in soluble form, and those are called antibodies. These antibodies can then bind specifically to the spikes on the surface of this particular virus and then dispose of it in a number of different ways that I will not describe today. But antibodies can only do battle with virus particles that are in your blood or extracellular spaces. They can do nothing to infected cells or the virus particles that they harbor. Now, some of these viral proteins that are made inside your cell will get chopped up into short peptides, about 10 amino acids long, and they are presented on the surface of these infected cells, not by themselves, but in complex with another large human protein called MHC proteins. Now, we each have 6 to 12 types of MHC proteins in us, but the MHC gene is the most polymorphic gene in the human population. That is to say, there are tens of thousands of variants in the human population. So that we might share between two people 1 or 2 MHC types, but the chance that two people who are not identical twins have exactly the same set of MHC molecules is about 0. Now, these different kinds of MHC proteins display different kinds of peptides from diverse viruses. And that I will become a bit more important a little later on. So that's the molecular flag of infection, a peptide derived from the virus bound to these MHC molecules. Now, T cells are also important players in adaptive immunity. Again, we have many clones of T cells, each with its own distinct receptor called the T cell receptor or the TCR. And if the TCR on this particular T cell can bind strongly to this particular viral peptide, it will get activated, proliferate, and then it can coordinate an immune response in a number of different ways. For example, certain kinds of T cells that are called CTLs are such that if an activated CTL sees another cell displaying the same peptide that originally activated it, it can secrete products that will kill the infected cell and all the virus particles that they harbor. Now, after you have cleared an infection, then most of the T cells and B cells that proliferated in response to this particular infection die off. Otherwise, your immune system would be primed for the infection that may not be your next one. But a few remain as so-called memory cells, which can mount robust and rapid responses upon reinfection with the same pathogen. This pathogen-specific immunological memory is the basis for all of vaccination. With a vaccine, what you try to do is induce memory T cells and antibodies that are specific for the bug against which you wish to protect the host. So as you might imagine, our interest in combating infectious disease and the untold amount of suffering caused by these kinds of autoimmune diseases that arise when adaptive immunity goes awry has led to a great deal of experimental research aimed toward understanding how adaptive immunity is regulated. And indeed, some spectacular discoveries have been made, especially over the last 50 years or so. But in spite of these major advances, an understanding of the principles that govern the emergence of a systemic immune or autoimmune response remains elusive. And just one practical consequence of this missing basic science is that over 30 years after the discovery of HIV, we still don't know how to make a vaccine against it. Other examples include malaria, TB, and so forth. So in my view, a major barrier to this quest for mechanistic principles in immunology is that the pertinent processes involve cooperative, dynamic events with many participating components that must act collectively for any given phenomenon to emerge. Moreover, these collective processes span a spectrum of scales. So when a single cell of the immune system recognizes a foreign pathogen, a myriad proteins inside this cell have to act in very precise non-equilibrium ways in order for the correct gene transcription of the programs to emerge. Many types of cells have to act cooperatively in tissues. And then, of course, there's the scale of the entire organism, and indeed populations of organisms that are evolving to try and beat our immune systems. So it is this what I call hierarchically organized cooperativity with feedback between the scales that often makes it difficult to intuit underlying mechanism from a few experimental observations. And further confounding intuition is that many of these processes are inherently stochastic in character. So over the last 15 years, we've been trying to help confront these challenges by bringing together approaches that have traditionally been stovepiped in different disciplines. In particular, what we have tried to do is bring together theory and computation that's rooted in physics and engineering with a diversity of different biological experiments and clinical data. A few words about the conceptual framework that we use a lot, which is statistical mechanics. If I were to be asked to give a one-sentence description of what statistical physics is, I would say that the central goal of statistical mechanics is to understand how fluctuating, interacting, microscopic objects can conspire together for coherent mesoscopic and macroscopic behavior to emerge. Given the challenges that I've just described to you that confront immunology, perhaps it would not be surprising to you that even though statistical mechanics and immunology developed on parallel tracks, i.e. with no intersections, us bringing it together has proven to be somewhat fruitful. Now, these experiments and clinical data are not carried out in our own laboratories, but with a diversity of different collaborators who are all immunologists, mostly at medical schools. Indeed, with the key assistance of these collaborators, indeed teachers, we've had some luck over the last few years in shedding light on a diversity of different questions in basic immunology and human disease that range from signaling in individual T cells that I alluded to, how this remarkable thing of a pathogen-specific T cell repertoire develops in humans where you are specific for pathogens that did not evolve before you were born, and also problems that confront the human immune response to HIV and vaccine design. And it is this scourge on this planet, and the work that I'll describe to you today pertains to this scourge on the planet. So my frequent trips to Africa over the last 6 years have made vivid the havoc that HIV/AIDS is wreaking in that part of the world. Just as an example, the area that I visit a lot is a province called KwaZulu-Natal, which is around the city of Durban in South Africa. And in this region, 60% roughly of women in their 20s are HIV positive. Coupled with the poverty that you see in those areas, it makes clear that a vaccine is urgently needed, and antiretroviral therapy is not the answer to the problems in these regions. A major barrier to the quest for a vaccine against HIV has been the high mutability of the virus. So in these phylogenetic trees, the number of N's corresponds to the diversity of circulating strains. As you can see in this diagram, which is to scale, the diversity of the flu virus in the entire world in a particular year is comparable to the diversity of HIV strains in a single infected individual, and completely dwarfed by the diversity of strains in just one region in Africa. So this high mutability of the virus is a major challenge for the development of vaccines because HIV can mutate to evade vaccine-induced antibodies and T cells that lie ready and waiting for a particular strain. This is also the reason that there is no known example of HIV being cleared by natural immune responses. So this challenge of designing a vaccine against HIV is truly a hard problem because you are asked to design vaccination strategies that will induce potent immune responses that do not arise naturally, or arise very seldom anyway. So then that brings up two questions. What are such potent immune responses, and how do we induce them by vaccination? So in the antibody world, partial answers to this first question are known because in the last 2 or 3 years, people like Dennis Burton and others have been able to obtain antibodies from patients which, at least in vitro, appear to be able to neutralize diverse HIV strains. These are called broadly neutralizing antibodies. And what points they target on the HIV envelope are known. But we have no idea how to induce these broadly neutralizing antibodies by vaccination. In the T cell world, an understanding of what's a good T cell response against HIV is beginning to emerge, but we certainly don't know even how to induce them by vaccination. So we work on both these questions, and I hope that at the end I'll have some time to tell you about some of our work pertinent to this first question. But first I'm going to focus on the second question, or as a metaphor, how to hit HIV where it hurts with T cells. So because of the high mutability of the virus, there has been interest in focusing a vaccine-induced immune response to target single residues of HIV proteins that appear highly conserved when you look at sequences of HIV proteins derived from virus samples extracted from diverse patients. The idea, of course, is that if you attack such a relatively conserved residue with an immune pressure from a T cell, then HIV would have to evolve a mutation there to escape the T cell pressure. But that should likely make the virus replicatively unfit, because why else do you see that residue relatively conserved in sequences? But this strategy has been of very limited efficacy, at least when you look at humans or monkeys that naturally target such residues with their immune response. Why? Because HIV can evolve other mutations, so-called compensatory mutations, that partially restore the fitness cost incurred by making the first mutation that evaded the immune response. So now you're more or less as fit as before, and you have evaded the immune response. Now, this way of thinking about virus vulnerability by just looking for single residues that appear to have a high or low mutational frequency really is incomplete because it does not account for these collective effects, such as these compensatory mutations. Many properties of an evolving virus population can be described pretty well by thinking of each residue as being independent. But if you're looking for virus vulnerability, then you need to define the collective compensatory pathways so that you avoid targeting those regions with a vaccine-induced immune response because HIV will evade them. And you need to define the combinations of mutations that the virus cannot make in order to target these regions so that HIV gets caught between a rock that is being killed by the immune pressure and a hard place that is making combinations of mutations which are deleterious for it. In short, if we really want to precisely define virus vulnerability, we need to obtain the fitness landscape of the virus. So in order to illustrate what I mean by a fitness landscape on a two-dimensional sheet of paper, consider a virus with just one protein with two residues. Otherwise, I can't illustrate it on this two-dimensional sheet of paper. On this axis, each grid point is a mutant amino acid that could arise in residue 1. And on this axis, each grid point is a mutant amino acid that could arise in residue 2. So any point in this two-dimensional plane is a particular viral strain for this simple cartoon virus. And on this axis is shown information about the fitness of that strain. So you can see how this information about fitness as a function of sequence will trace out hills and valleys, but of course in a much more high-dimensional space than I've shown over here. Now, knowledge of this fitness landscape can be very useful for vaccine design because with a vaccine-induced immune response, you want to push the virus off of one of these fitness hills and force it to make combinations of mutations that correspond to one of these fitness valleys. And you also want to block these mountain passes that correspond to these compensatory pathways that HIV uses to go from one hill to another when this one is under immune attack. Knowledge of this fitness landscape can then really inform the design of the active component of a vaccine. Why? Because instead of the vaccine's active component being the whole bug, as is usual, you can choose only regions of the proteins of the virus in ways that satisfy three different criteria. One is that they minimize regions where compensatory pathways are more likely, because you know these collective effects. And you maximize regions where multiple mutations are especially deleterious. That way, the immune response that will develop will be for these regions, and hence, it will force the virus to make mutations that it doesn't like to make. And finally, as I told you, different MHC molecules present different kinds of peptides. So you just can't design it based on these two criteria because if no humans, or a very small fraction of humans, can present peptides from these regions, it won't matter. So you also have to maximize regions that can be presented by people with diverse genotypes or different types of MHCs. So it is for these reasons that we began to think about developing a fitness landscape for HIV and then exploiting it for vaccine design. So there are many ways you could try to do this. One way you could try to do this is by so-called mutational scanning experiments. You make lots of mutations and you measure the replicative fitness of these viral strains in vitro, and then you go to your neighboring friend in machine learning and computer science and ask him or her to learn this landscape for you. In the long run, maybe this will be possible. But today, given where we are in what can be done with mutational scanning experiments, you can largely probe only single residue mutations because the combinations of mutations are too many. You just think about one 500 residue protein and imagine that each residue can only be wild type or mutant, all simplifications of course. Still you have 2 to the 500 possibilities, which is 10 to the 150, which is a humongous number. So rather, we thought that another way to think about obtaining these fitness landscapes is that the sequences of viruses derived from diverse patients contain information on the fitness landscape. So can we go from these available sequences to this kind of fitness landscape? And this is something we've been working on for a number of different years with a number of different people. These are the people who are no longer here in my lab. Andy Ferguson, Vincent Dahirel, and Elizabeth Reed are all faculty members of their own independence now. And Andre Kosmarel and Karthik are postdocs. And much of the work that I'll describe today is a collaboration with Bruce Walker's lab. Bruce is a physician scientist at the Massachusetts General Hospital. So I'm not going to tell you all this historical work because I've spoken about them before at HKUST. What I'm going to do rather is tell you more of our current work, largely unpublished. So our current thinking goes something like this. What we really want to do is the following. The sequence data contains, so any given sequence can be considered to be a vector Z. Why? Because it's just a list of different amino acids at different positions. The sequence data contains information on the probability of observing a particular sequence. But this sequence data also contains information on the probability of single mutations at every residue, double mutations at every pair of residues, and triple mutations at every triplet of residues, and so forth. So any mathematical model that reproduces these mutational correlation functions is a legitimate model for P of C. So what we did was we took a page out of work that Bill Bialek had done in the context of neuroscience, actually, firing of neurons and so forth, and we asked, what is the least biased model for P of Z that by construction fits the 1 and 2 point mutational correlation functions? Least biased has a name mathematically, and these are called maximum entropy models. So to illustrate that, let me go through the mathematics of this for just a bit. Any given sequence k can be represented as a vector zk, and for simply illustrating the mathematics, let me consider a situation where only two kinds of amino acids exist at any given residue. Generalization of this to multiple colors of residues and amino acids is trivial. And obviously we've done that. This is just to illustrate what I'm proposing here. So P of Z is the probability distribution of observing sequences. That is what you first seek. And P of Z log P is the entropy of that probability distribution with a negative sign. You want to maximize this. Why? Because that will make it the least biased distribution. But you want to do it with constraints. The probability distribution should be normalized. The probability distribution for 1 point mutations predicted by the model should be those that are observed. And the prediction for the frequency of double mutations that the model predicts should be that's observed. And you enforce these by these 3 different Lagrange multipliers, which is standard way in mathematics of putting constraints to optimization problems. 2 lines of algebra of differentiating with the probability of Z will tell you that the probability of observing sequences is given by this sort of Hamiltonian form, which is very, very common in statistical mechanics, with this Hamiltonian having a form like this, with the fields HI and the couplings JIJ are such that the 1 and 2 point correlations are satisfied. That is, they are what they are observed in the sequence data. I should also point out that because these coupling constants JIJ can be both positive and negative, this Hamiltonian has a structure very similar to spin glasses and to that of Hopfield's model of neural networks. And I shall come back to that analogy a bit later in this talk. Now, another important problem is that this P of Z is really a model for the prevalence of sequences. You might imagine that the more prevalent sequences are also more fit. That's why you see them more frequently. But this relationship between prevalence and fitness could be significantly complicated. Why? Because the sequences that we are using to infer this prevalence landscape, and fitness landscape if you like, are not viruses growing in petri dishes. They are viruses extracted from humans where there has been a massive non-equilibrium battle between the immune system and that virus. So in order to explore this relationship between prevalence and fitness, we carried out first a lot of theoretical analysis. I shall, of course, very soon compare this with real in vitro and clinical data in just a second that we predicted. But for the moment, let me just quickly take you through some of the mathematics of this. And those of you who don't want to just listen to it, I'll tell you the main result before we start the few slides on the mathematics. Anyway, this work was done with my physics colleague Mehran Kardar at MIT and the aforementioned Karthik Shekhar. The main result that we have is that the rank order of fitness and the rank order of prevalence is the same. The fitness and prevalence are not the same, but the rank order is the same. And this is true for HIV for a very important reason. HIV has not evolved in response to directed immune pressures, either natural or vaccine-induced, that have been effective. So the evolution of HIV, unlike, for example, flu, has not been specifically directed in different directions at the population level. In each person, of course, the immune response is a big deal. It is directing selection within a person. But when you infect another person, there's a huge diversity of genetics because of reasons I've told you. And so different people attack different regions. And if what happened in the past person lowered the fitness, it reverts. And so at a population level of viruses, the immune response is basically just a perturbation. It is not a perturbation in each individual, but it's a perturbation overall. That's the reason. Well, let me take you through the arithmetic that got us there. So first, Karthik did a few simulations that were aimed to mimic the way that these virus samples were collected. So he would infect a computer patient with some virus, and a host-pathogen dynamics would ensue here. I'll show you what the dynamics was in just a minute. Then randomly, after some point in time, he would collect a sequence from this person, and he would infect another person. And then a different host pathogen dynamics would ensue because genetically this person is likely to be different. And this process would continue. And at randomly chosen times, he would draw a virus sample from this patient, and then he would put that in his data bank, because that's roughly how this sequence database was collected. So what are the dynamics in each person? So the fitness, or in our description, the Hamiltonian of statistical mechanics, is divided into two parts. One is an intrinsic fitness, and the other part is an immune pressure that acts in this person at particular sites, at only a few sites in each person, which promotes mutations at those residues because the virus wants to escape the immune pressure. The dynamics is actually very simple. You mutate every sequence with probability mu per site. That's the probability with which virus HIV makes mutations, 3 times 10 to the minus 5 per replication cycle per base pair. You then kill the sequences with probability proportional to their fitness or lack of fitness, and you just keep repeating this. That's the dynamics within each person, and you choose this differently, which sites are being attacked in different patients because of genetic diversity, and this distribution is chosen based on clinical data. Now we assume that the fitness landscape, the intrinsic fitness, is the fitness that we infer from the prevalence of the sequences. Why? I'll tell you in just a minute. So at the end of the day, you have two sets of sequences. A set of sequences that came out of Karthik's computer simulations, and a set of simulations that were in the real database. I assert that if the mutational correlations in both sets of sequences are identical, then our procedure of just using the sequence database to infer the prevalence landscape gives you the fitness landscape. Why? Because we know that the real sequences, by construction, the mutational correlations in the real sequences will be obeyed by this intrinsic fitness model. So after doing all the dynamics and these mutational correlations are the same, we'll infer the same thing as what we assumed was the intrinsic fitness. Are they the same? Well, his computer simulations show that they're not the same. So this is in the real data, this is in the dynamics, they're not the same. 2-point, this is the 3-point, they're not the same. But they're monotonically correlated. They're not random. By the way, if our Hamiltonian was a ferromagnet of statistical mechanics, we would be done now, because there's something called the Griffiths theorem, which says that, if the mutational correlation function or the correlation functions are monotonically correlated, then the coupling constants are monotonically correlated. That is, all that will happen is that the whole fitness landscape will move up and down. There'll be a uniform up and down shift. But we don't have a ferromagnet. We have an Ising spin glass. We have a half-field model. So there is no Griffiths theorem for these kinds of models. So what do you do? So we did what old men know how to do still, analytical theory. So this is Eigen's famous equation for viral evolution. This is xi is the fraction of viral strains of sequence I. This is its time derivative. This is the replicative fitness of that virus. So this is replicating. These are mutations to other virus strains. These are mutations from other viral strains into it. And this strange-looking term you see over here is necessary for xi to be a fraction that is normalized. And if you think about what it really is, this is the average fitness of all the viral strains. And all it's saying is that your fitness has to be compared with your peers because it's a competition. There's a guy called Lauthauser published a paper a long time ago in JCP, of all places, where he showed that this non-equilibrium dynamical equation is isomorphic with the equilibrium statistical physics of a two-dimensional Ising model, with each row being a virus, the row below it being its progeny. And he derived a Hamiltonian that corresponded to this, where the in-row couplings were just adapted to our problem, is the intrinsic fitness and the immune pressure. And across rows, there is a ferromagnetic coupling which simply reflects the fact that if I am a particular amino acid here in my parent, my child is likely to have the same amino acid there because the mutational frequency, even for HIV, is much less than 1. This Hamiltonian gives you the statistical probability of seeing a particular configuration, and a particular configuration is a particular evolutionary trajectory. So it's a Feynman path integral approach, if you like. Once you have a Hamiltonian, you can do all kinds of tricks that were well known in statistical physics. For example, you can say this is the real Hamiltonian, this is the Hamiltonian that I infer. How good can I do with this form of the Hamiltonian? And you can use ideas that Feynman developed in the context of polarons and so on and so forth to actually figure this out. And this is how we saw that the prevalence and the fitness are not exactly the same, but their rank order is the same. As you might imagine, my immunologist friends and clinical collaborators were not at all impressed with these hieroglyphics and ancient Greek symbols, and they said, well, show me the money. And the money really is comparison of predictions that you make for replicative fitness of viral strains using your model and in vitro measurements of the same species. So the metric of fitness in our model is a quantity called E. So you give me a sequence, we will return a value of E. And the theory would predict that E of a mutant sequence minus E of the wild type should correlate negatively with the logarithm of the fitness of the mutant strain divided by the fitness of the wild type. So I'll show you today predictions for 35 different strains of multiple, single, double, triple mutant strains that we made. Bruce Walker and his friends Tumbi Udungu in South Africa made these mutant strains. They measured their replicative fitness in vitro, and here is the comparison. So that's the theory, that's the experiment, and only 26 different strains are shown here because for 9 different strains we had predicted that replicative fitness should be essentially 0, and they did not grow out. I did not put them on here because the difficulty of plotting the log of 0. But as you can see, we are not perfect by any means. But in all the things we have tested so far, you capture large fraction of you capture about 82% or 80% of the variation. I should also point out, in older work that I've described here before, we have also, in many cases, been able to understand the structural basis for why certain combinations of mutations are really deleterious. And the interesting thing we observed there, that is not because of the structure of a single protein. You would never intuit it by looking at the structure of a single protein, but because of the complexity of making multi-protein assemblies that are critical for viral function, and many interfaces get disrupted when you make these combinations of mutations. We have also found that the combinations of mutations that we predict to be exceptionally deleterious are also exactly the regions of the virus that are attacked by natural immune responses by a group of patients that Bruce discovered called elite controllers, whose immune response naturally keeps the virus at low levels. We've also been, as I say, in addition to these elite controllers, we've also been testing more recently our ability to predict viral evolution and fitness in individual clinical patients with Andrew McMichael's lab in Oxford. And this work's done with current postdoc John Barton. So what Andrew has, again, I'll be relatively brief. What Andrew has is that he has a cohort of patients in which he follows the evolution of the virus by sequencing the virus at different periods of time. And he also takes blood from these people and measures what is the immune response that they are putting, which regions are they attacking. So the first question he asked is the following. In each of these patients, different mutations could emerge as escaping the immune response because the immune response is targeting multiple places. He said, can you tell me of all the possibilities where escape could occur, where will escape occur? So I'm not going to show you the data here, but we can predict where the escape occurs with about 85% accuracy. And I want to emphasize one thing. They had a very poor record of predicting these things just by looking at conservation frequency. And the reason is our results highlight the fact that if different possibilities are there, the compensatory pathways will be used, where you make multiple mutations to compensate. And furthermore, the sequence background that the patient gets from the other patient matters a lot. If the right background is present, escape will occur much faster because the compensatory mutations already exist. The second question he asked is, can you rank what are the times at which escape occurs in these individual patients? Can you tell me that escape will occur first in this guy, second in that guy, third in that guy, and so forth? So as you know, these kinds of evolutionary dynamics, such as the one I showed you that Karthik did with the toy models before, have been done for a century, ever since Sewall Wright and Fisher. But they've always been done with fitness landscapes that are toy models, one peak, 2 peaks, things like that. So we think this may be the first evolutionary dynamics done with the fitness landscape of a real virus. So here's the evolutionary dynamics using Wright-Fisher-like models predicts evolutionary generations of evolution, not real time. So this is our prediction in generations of evolution, and this is the escape time that Andrew has measured in these real patients. Again, you see we are not perfect, but again, about 85% of the data, we get the variance. Now please note, these are not contrived experiments. Each dot there is a real human who has been infected and which you're watching escape. So we're a little bit excited about these results if they continue. People at Hong Kong University are trying to do these things for the flu now and so forth. But we're a little bit excited because it's possible that modifications of this idea can give you a way to combine two technologies that are both becoming faster and faster, sequencing and computation, to extract fitness landscapes of viruses and do rational vaccine design rather than what Jenner and Pasteur have been and people have been doing since then. So actually we have designed an immunogen for a therapeutic vaccine, T-cell based vaccine against HIV using the ideas we have developed over the last several years. And these are just long peptides that have to be hooked together. They follow those 3 criteria that I said at first. The difficulty with such immunogens that are just long peptides hooked together is that they're not very immunogenic when delivered in the usual ways in which full vaccines are delivered. So we are collaborating, we are collaborating only in spirit in these things, but several approaches are being used. One approach is being used by my colleague Darrell Irvine at MIT, who has developed nanoparticles where one end of the whole deal gets you to the lymph nodes, our immunogen has been synthesized into it, and it has other things called molecular adjuvants. And in the context of cancer vaccines, at least in mice, his lab has shown that you can get a big T cell response. But mice are not humans, and mice are not monkeys, and monkeys are not humans either. So much optimization has to be done. And Darrell is currently collaborating with Dan Baruch at Harvard Medical School. And they have actually injected these into monkeys. And the first and second boosts of this have shown that they do get a response, but it's much weaker than they would like it to be. So there is more optimization to be done, which they are doing, along with Uli von Andrian, another Harvard Medical School person. But if this optimization of the delivery method can be done, what Bruce wants to do is he wants to take this and put it into infected patients who are currently under antiretroviral therapy. See if you can redirect the immune response in ways that target the vulnerable bits. Take them off of ARVs and see if you can convert them into these so-called elite controllers who will control the virus forever. Probably never work. But there is some hope, because there are these guys in EPFL who took our older papers, not our new design, our older papers where we had first shown some of these vulnerable bits. They took those bits and they combined it with an adjuvant, which these three people claim is a lousy delivery method, but it is FDA approved, and they put it into a small cohort of patients. And they published this in Lancet this year, where they showed that you didn't get this huge drop in viral load that we want, but they got a drop in viral load in those who are vaccinated by this. So the hope is that with better delivery method and our new improved immunogen, perhaps we will be able to do this. So far I've presented all of my work as sort of directed to this very practical goal of a vaccine. I want to tell you a little bit about how some of our work in getting the fitness landscape of HIV is also showing us some interesting basic connections between HIV evolution and other branches of inquiry. So we have a fitness landscape. So what we can do is we can take every sequence of HIV, that protein that is known, and we can subject it to a zero temperature Monte Carlo using our Hamiltonian. That's fancy language for saying take every sequence and keep making mutations until you keep climbing the hill and get to the top and you don't change anymore. Okay? Now I will call these hills attractors or basins for reasons because it connects better to statistical mechanics and neural networks. And I'll make those analogies in just a bit. What we found is that for all the proteins in HIV, all these thousands of sequences would partition into a few of these fitness hills or basins. Okay, this is a schematic. Then you can do the following. You can go to every basin and say, count the number of sequences in each basin. And then you rank order the basins with the most populous basin being ranked 1. Got the idea? And then you make a plot that Ziff made 100 years ago for the first time in the context of linguistics. You make a plot where on one axis you plot the number of sequences that fell into that basin, and on this axis you plot the rank of that basin. This is what you see for a large polyprotein of HIV called GAG. This is a log-log plot, so you see it's a power law. Here's what you see for NEF, another polyprotein of HIV, a power law. Power laws are special because, as you know, what power laws mean is you have very fat tails to the distribution, and so what they're saying is no typical number of sequences that fall into any basin. Incidentally, if you read a book, let's say Moby Dick, and every time you encountered a word, you put it into a bin, and at the end of the day, you counted out how many times you used a word, and then you rank ordered the words according to usage, with the most frequently used word being ranked 1, and you made this Ziff plot, which Ziff first made, you would see the power law like this. But we don't see this for all HIV proteins, though. Here's protease, another HIV protein, no power law. We don't see it for integrase either, another HIV protein. What's the difference between the ones for which we see power laws and for the ones for which we don't see power laws? Well, GAG and NEF are very immunogenic. That is, they attract large amounts of immune pressure from humans. Protease and integrase are not. They get hardly any immune response from people. So what does all this mean? So here's what we think it means. So consider the immunogenic proteins first. There is no question that immune responses force exploration of sequence space by forcing mutations to evade the immune response. And we can see this because if we go and look at the sequences that lie at the top of the hill or the bottom of the valley, the ones that they all go into, they are highly enriched in mutations that are known to be associated with human immune spots of attack. But these mutations to avoid the immune response always lead to fitness cause. The virus has to make compensatory mutations to be fit enough that you see it in the population. How do we know that? Again, if you go look at the basin sequences, they are enriched in mutations which, according to our fitness landscape, have positive couplings between them. That is, they are compensatory mutations. So it is this collective effect of mutations that are positively coupled or compensatory that define the basins. That's the collective effect that's defining it. Now, the fact that we have these attractors is not surprising because our Hamiltonian has a form similar to Hopfield's neural network model, which has these basins. In fact, in that case, these basins are considered to be stored memories of past stimulation. Our basins are also stored memories. They are stored memories in the HIV population of the compensatory collective mutational pathways that HIV used to evade diversity of different human immune responses. Now, why is there a power law or no typical population size of basins? We have a great diversity of immune responses, so there is no typical immune response. Which means that every time an immune response is encountered, a different type of a class of immune responses is encountered, a different type of mutation has to emerge. Different sets of compensatory mutations have to be made. These compensatory mutations are constraints, and different numbers of sequences are consistent with different constraints, and there's a whole diversity of constraints, so there's a whole diversity of number of sequences that fall into it. For the non-immunogenic protein, none of this is true because, there's hardly, it doesn't push the sequences around. You have one or two basins that have almost all the sequences and the rest of them are not. So you won't get power law, you'll get a sharp exponential decline. One can make all this more precise, and how much time do I have? Well, so let me not tell you about how one can make a little mathematical model to describe this. That'll just tell you that we can. It's a detail that if someone's interested, I can tell you after the talk. Let me skip that, and let me take a few minutes though to tell you about some of our work on antibodies, which is the right amount of time is left for that, because I'll just give you a 30,000-foot view of it. So first of all, how do antibodies develop? In my introductory slide, I said that here's what happens. The B cell gets activated, and then somehow magically antibodies emerge, that is, the B cell receptor gets secreted. Actually, something much more interesting happens. When the B cell gets activated, it seeds a structure in your lymph nodes called the germinal center. In this structure, a Darwinian evolution process occurs in a very short time. The activated B cell begins to proliferate, but as it proliferates, it starts to introduce mutations into the receptor at a very high rate. It's called hypermutation. So you get lots of mutant B cells. These mutant B cells then transit over to another part of this germinal center, and there they encounter the virus that originally activated this, displayed on other cells, and the B cells compete to bind to this virus. The ones that bind more strongly eat more virus because they engulf the whole thing with the receptor. And then there are T cells, and then they take these things, chop them up into peptides, and display them. And then they have to get a second signal by interacting with the T cells with the T cell receptor. And the guy that ate more virus has a better shot of outcompeting their neighbor in getting this survival signal. The others die. So you see this is a Darwinian evolution process that occurs where every time you're selecting for B cells with receptors that bind more strongly to the virus. Now, most of these guys are then recycled back for more rounds of mutation and selection, and a few of them get out and secrete these antibodies. So you see, with time, you will get higher and higher affinity antibodies as the infection is controlled. So this process is called affinity maturation. And ever since the papers in 1964 and 1963 from Hermann Eisen, one of my great collaborators and friends who passed away 10 days ago at MIT, 96 years old, was working on a paper the day he died. Okay, and then lots of people since then, and this is the most well-studied problem in immunology via computation. So I never thought I'd ever work on this problem. I really believed this was a closed problem. But then I started listening to these people giving talks about how do I induce broadly neutralizing antibodies by vaccination. You know, I told you about these broadly neutralizing antibodies that neutralize diverse strains. So it's very clear that you're not going to get an antibody that neutralizes diverse strains by putting as the vaccine one strain. If you put one strain, this process that I described to you will give you high affinity for that strain, because that's what the system is doing. It's Darwinian evolution. So you'll have to put in multiple strains for sure in order to get antibodies that bind strongly to more than one strain. That immediately raises the question, what should these antigens be? But even if you knew them, there are other questions. What should be the temporal pattern in which you give these antigen variants? Now the variants will be as follows. They will share some conserved residues that say HIV has, but they will have variant mutations around it. So which temporal pattern should I administer them? What should be their concentrations? And I just listened to these talks and I said, why don't they go and look at the knowledge about affinity maturation and figure out how to guide these choices, because the space of possibilities for this is so large that you cannot really comprehensively sample this with experiments in monkeys. Well, here's the challenge. Every study of affinity maturation has been done with affinity maturation induced by a single antigen, never with multiple variant antigens. So that's why we think that if one had a fundamental understanding of how affinity maturation occurs when there are multiple variant antigens, you could focus experiments on a few productive choices, but more importantly, you would also get a fundamental understanding of a very basic question in biology. So I'm going to focus today, and I'm going to again say, I'll give you a 30,000-foot view of this, of just what should be the temporal patterns of antigens in order to guide this evolutionary process to get broadly neutralizing antibodies. And to answer questions like that, we've developed a computational model of affinity maturation. And this work was, again, done in collaboration with my physics friend, Maran Kardar. And the postdoc who led this work is Shenshen Wang, who incidentally got her undergraduate degree in physics here at HKUST, and then a PhD in physics from San Diego, and a postdoc with me at the current time. I won't tell you what we did for the model, because time is running late. But let me just show you some results that are intriguing. So we're going to use 3 different kinds of antigens, a wild-type antigen and 2 mutant strains that have non-overlapping mutations. And we're going to look at 3 different schemes. Scheme 1 will administer all 3 as a cocktail. Scheme 2 will give 1, and then the cocktail of the 2 remaining. And scheme 3 will be sequential. And each time what we do is we run this calculation many, many times. And we look at which antibodies come out of these germinal centers, or GCs, and then we scan them for affinity against a standard panel that is used in all the experiments to test whether there's neutralization or not. And in this way, we determine, in each case, how broad was each antibody, how many variants of the real virus was it actually neutralizing. So we get the breadth of each antibody, and then because we do many, many calculations, we can look at the probability of obtaining a different kinds of breadths. So here it is, what happens when we immunize with a cocktail. So here is the histogram from this is from computer simulations, this is the probability of seeing a particular breadth of an antibody, and 1 is full coverage, and you can guess the rest. And you see in this case that you don't get any broadly neutralizing antibodies. Moreover, we find that very few GCs succeed. Most of them just collapse. Most B cells die. We call this situation frustrated affinity maturation for reasons that are related to physics. But you can see the problem. you have, let's say, 3 antigens. Early stages of affinity maturation, there is no strong interactions with anything. Not the conserved residues they share, not with anything. But a B cell gets selected by one in one round, mutates, comes back, will likely see a different one. Won't bind to this because it doesn't have strong interactions with the conserved core. It's going to apoptose. So this is what is frustrated affinity maturation. And it's a very interesting problem that I want to do a really analytical general theory for, because this is evolution driven by potentially conflicting selection forces. Sometimes you see this, sometimes you see that, and you have to somehow navigate this. So this is not a good strategy for vaccination. Here's what happens: you give one guy, and then you give a mixture after that. 2 stages. Again, probability as a function of breadth. Now you do see some broadly neutralizing antibodies, but with low probability. But you also start seeing that antibodies are produced now. This is what I think happens during natural infection, because you get infected with one strain, then it diversifies. And with very low probability, some patients make good broadly neutralizing antibodies. And that's what you're seeing. There's a lot of effort to try and understand how these patients made the broadly neutralizing antibodies. Good thing to do. But it may not be the best vaccination strategy, because you don't want 3% of the people to succeed or 7% of the people to succeed. Why is it better than the first case? Because in the first round of affinity maturation, you got some strong interactions with the conserved residues. So the frustration became less when you gave the cocktail. You were more able to negotiate this conflicting selection forces. Here's sequential. Now you see broadly neutralizing antibodies. And the reason, if someone is interested, I can tell you, but basic idea is in the first round you get strength with the conserved residues. You give a second variant which has some mutations. There are 2 types of lineages that can evolve. One which increases strength with the mutations now. The other, because it has strength of binding to the conserved residues, just removes the interactions with the mutant sites. Then you give a third variant that has mutations elsewhere. The first kind, which increased strength of interactions with the mutations, now is screwed. That lineage is likely to die. The other one simply takes the ones off with the other one. And so it just focuses down the core. So let me just close by saying that these have been tested now in mice. So they made 4 monomeric GP120 variants, which are HIV envelope. And they then asked which temporal order focuses the response on the CD4 binding site. The CD4 binding site is the conserved residues. And they have mutations all around them. So this is what happens when you give a cocktail. These are the antibodies that are made, and they're binding to the 4 variants. So the higher here, the more the binding. And you can see that for the 4 variants, they have wide distribution. It doesn't bind to this at all, a little bit to this and some to that. For mice that were sequentially immunized, you see a much narrower distribution of these things. So they've done a bunch of other experiments to show that you really do get focusing on the conserved residues by giving it sequentially and not giving it as a cocktail. But these are mice. These are monomers of GP120. But they are true for even the monoclonals. So anyway, what happening now is that Dennis is going to put these in, Dennis Burton, who discovered some of these broadly neutralizing antibodies, is going to put them in monkeys with some antigenic strain and see whether similar results can be replicated in monkeys. But we are very early days in this whole project. So with that, I'll stop. I thank you for your attention, and I apologize for the slight lateness of the hour.
So any questions from the audience? We have expert in neuroscience.
Well, I was going to ask a question on the very last part. There's the sequential introduction of the mutant strains has to be timed relative to a certain internal dynamic that's implicit in your model. So tell me about, so it has to have something to do with the temporal dynamics with which B cells are going to recruit plasma B. Somewhere in there's a critical time calibration you have to do.
So this, basically, it takes about 2 weeks or so for any one cycle to complete. And so you have to give it at least after that time. And you don't want to wait too much after that, because if you wait too much after that, a lot of these cells will die off.
Then it'd be as if you had given just one.
That's correct. So that's the timing of the thing. And then there's other questions here that are very deep. I mean, what should be the concentrations? So actually, the concentration can't be too low because nothing will happen. The concentration is too high, you'll just get strain-specific antibodies because anything will get selected. So actually, I think, although I can't give you a precise answer to this question, this degree of frustration is influenced by concentration, kinds of antigens, how many mutations are they separated, temporal pattern, and other variables. And what you have to do is choose the variables to optimize the frustration. Why? Too little frustration, you'll get strain-specific antibodies. Too much frustration, you'll get nothing. So have enough tension to make the system want to do this, not too much that it leads to apoptosis.
Yeah, so I found it surprising that you got such a big signal with a sequential of just 3. Such a big signal of broadly responding antibodies with just 3.
Right, but in the computations, these 3 were chosen in a very precise way, that there were 11 non-overlapping mutations.
There's 0 overlap.
With 0 overlap between the 22. So that's why.
So it couldn't be just any 3 random.
No, it cannot be anything random, and that's why it's very important that the immunogen be designed properly and be the trimeric immunogen. For the monomer, that the mice experiments, they did a similar strategy. We took the 43 sites around it and then made mutations around them. But now you have to do that for the trimer, which is harder. Absolutely.
Any other questions? So I have a question also on the second part. So are you suggesting that we don't really need to design broadly neutralizing antibodies, we're just using a dosage strategy? Then you can actually have a broadly neutralizing effect. The antibody is not that we suggested here are not actually broadly neutralizing.
No, they are. They are broadly neutralizing. I'm suggesting that there are different mechanisms, ways in which you can vaccinate to make this Darwinian evolution generate broadly neutralizing antibodies, and most methods will not do it. So I'm saying the sequential has the best shot. But I'll tell you something. This is a very hard problem, and I really think it will be very hard to experimentally actually do this because the design of the antigen, the question that you asked. But my assertion is that if there's a chance, sequential is the best chance, not mimicking what happened in natural infection.
My second question is on the power scaling law, and we see the gap proteins, and you make a point about this protease integrase why they doesn't have this grand order scalings. But how about if we look at the HA on influenza viruses?
I don't know.
Because in the HIV, I guess those information is stored. But HA, because the antibody is always more like mutate through the antibody.
Yeah, it may be more directed and may have a different response. I do not know this.
Okay. So Ray, did you see any scaling of?
No, I haven't looked into that, but that's definitely something I should look into.
But you must look into it after you do it with your sparsity matrix in place.
Right.
Okay, because otherwise you won't get you'll get such a rugged landscape. You'll get thousands of little valleys.
Maybe that's the reason why our results didn't match up.
Exactly. I mean, I've thought about it. At that level, I've thought about it.
Can I ask a follow-up question on the zip floor? So, in that, so you found a lot of sequences which belong to different basins in your zip floor plot. So, is there any significance between sequences belonging to the same basin? Like, are they related in some way? For example, through like same MHC, for example.
They are related by the fact that they don't share exactly the same MHC, because different MHCs can present similar peptides as well. But they are related by the fact that they have a core of mutations that are compensatory in the same general vicinity of attack.
But does that imply that, like, maybe this...
So it's the same class of compensatory pathways.
But is it true that in the same kind of cluster, the same basin, you have a group of MHCs, which might be different from another group. So doesn't the MHCs kind of drive which sites are mutating due to compensatory mutations?
Correct. But you can't separate the MHCs perfectly. We looked into this. We cannot separate the MHCs exactly because each human has more than one MHC. So you're seeing some concatenation of them, and whichever one dominates at a particular time. So rather, I think it's better to just think of it as to which mutation was driven and what compensatory mutations had to get made. So that's the way we're looking at it right now. But I do find it interesting that you could, with analogy to the kinds of things that the Hopfield did and so forth, you can think of this as memories in the HIV population that are stored of the complex ways in which it escaped. Because now it also, I mean, I don't know, right? Presenting, I don't know how it is useful. It's cute for sure, but I don't know if it's useful. If it's useful, it may be because you can now look for what we did by just looking at the fitness landscape, but perhaps in this lower dimensional representation, you can now look for what are the complements, that is, what mutations combinations cannot make by just looking at these, which we did before in a brute force way.
Any other questions? I see some food outside. I guess it's for us, right? So I guess then we can stop here and have some food for refreshment. Thank you very much for your